Good afternoon. I would like to welcome you to today's Network Verification Challenge webinar entitled BEL Tutorial. At this time, I would present today's speaker, Brett Fields. Brett has been with Solventa since June 2012, during which time he served an instrumental role in constructing the biological networks being reviewed during the Network Verification Challenge. Brett, please go ahead.
Okay, great. Thank you, and thanks everyone for joining us for the webinar today. So our first webinar in the series will cover the BEL language, which was used to construct the networks that are available on the network verification website. And hopefully today's tutorial will give more background in terms of how to review edges and how to add additional evidences and edges as you come across these in the networks. Okay, so a general question to pose in the beginning is essentially why learn the BEL language? So here you see a generalized representation of what the networks appear as on the NVC website. And so a large part of participating in the challenge is to review the BEL encoded edges and nodes that underlie the biological networks on the website. And an additional feature that you could use the final versions of the NVC networks for your own research. And so a better understanding of the BEL language will enable you to capture these final versions of the networks and then apply these to your own research findings. And also a general knowledge of the BEL language will enable you to capture, store, and share your own scientific research with the international BEL community. So the BEL language has a very large and growing user base, and most of the findings that other labs are contributing can be found on the openbel.org website. And so a better understanding of the language in general will enable you to better share your own scientific findings with the larger scientific community. Okay, so one general feature of the BEL language is that it essentially enables you to capture scientific findings in a human-readable but also computable format. And so here's a general overview of how statements are constructed. So first, you start with the scientific literature of interest. So these can be papers that you acquire from PubMed. And within these studies, you can find specific statements that link the biological activities of different proteins together. So in this one example, it would be the RNA expression of RBL2 is directly mediated by the activation of the FOXO3 transcription factor. And then here underneath, we see the original citation of what journal it was published in. And if you move further and translate this into the BEL language, we see that this is a very readable format, so it's easy to understand that the transcriptional activity of the FOXO3 protein is responsible for directly increasing the RNA expression of the RBL2. And so this general format enables you to very easily capture findings as you find those in the literature into the BEL language. Okay, and another feature to touch on is what specifically differentiates the BEL language from other languages that have been used for network generation. So generally the BEL language represents discrete scientific findings in a computable form, but it also enables you to capture qualitative causal relationships and also attach to this the relevant contextual information that underlies those relationships. And I'll go into several examples in the following slides that give more background here. And another feature is that BEL can drive knowledge-based analytics. And so encoding scientific findings in the BEL language really enables you to apply more quantitative metrics and use these for network scoring for microarray data. And then another feature is that in other languages, such as the BioPax and SBML languages, are primarily designed with pathway construction and visualization in mind. But at the same time, the focus is largely on very detailed molecular events, such as capturing different phosphorylation states of AKT1 and precisely what mediates those changes. And this is really opposed to the multi-level pathways that can be captured within the BEL language that include up to the general process level. And so we'll see a few examples of how this translates to the language in the following few slides. And another feature is that the other languages such as biopacks do not enable you to capture contextual information for the findings. And so when we refer to context here, we basically just mean what species or what specific cell types these scientific findings were originally generated in. And so these other languages don't have this capability, but you are able to capture this additional information in the BEL language. Okay, so here is just a graphical representation of a few examples of what you may see in the BEL language on the left side and biopacks on the right side. And so similarly to biopacks, BEL also captures pathway information, but as I mentioned, it also includes the causal relationships that are backed by discrete scientific findings. And this also includes contextual information. And so to highlight these in more detail, we see on the right side the biopacks, a network constructed using the biopacks language. And essentially you can see a very granular level of detail between nodes connecting in the network. And so at the top here, we see the AKT1 protein, and this has potential phosphorylation sites indicated on the right side as a circle and residue 308, and then also residue 473. And basically the flow through the structure is just to illustrate how these AKT1 isoforms can be interchanged between phosphorylation states and there are a few proteins that mediate this process indicated on the left side. And so one example being PDK1 inhibits the switching from the AKT1 unphosphorylated isoform to the AKT1 phosphorylated at residue 308. And so this is an extremely granular level of detail that really doesn't capture contextual information. And so one example being in this interconversion event between AKT1 unphosphorylated and phosphorylated at 308, we see that the PDK1 protein could be responsible for that, but it could be the case that we're interested in this type of relationship in a cell type that doesn't express PDK1. And so this type of contextual information really gives more meaning to the structure of the network. And basically this is what BEL enables you to do in the network. And so moving on to the BEL example here on the left-hand panel, we see a very generalized representation of what you may see in a BEL constructed network and how the nodes are interconnected. And so on the top here, we have the kinase activity of the IRAK4 protein, and this leads to an increase in the kinase activity of the AKT1 protein. And then this downstream leads to increased catalytic activity of the NADPH oxidase complex. And if we zoom in further detail on the BEL network here, we can see that within the structure of the network, we also capture specific contextual information related to this edge relationship. And so in this example of the kinase activity of IRAK4 increases the kinase activity of AKT1, we see that this has a supporting publication indicated by the PubMed ID here, so 174758888. And this also contains contextual information such as it being from a mouse, as well as this specific finding was captured in a neutrophil context. And so this really gives more depth to the networks and enables us to better represent full biological pathways and how those lead to phenotypic level events using the BEL language. And then another feature of BEL is also that it's capable of driving analytics platforms. And so when we can say the language is computable, it simply means that the networks that have been constructed using the BEL language, these can be scored using omics data to facilitate an enhanced and systems level understanding of the processes affected by perturbation. And so here in the graphic at the bottom portion of the slide, we see a series of perturbations that are possible in an experimental context. And so this could range from mouse knockout to a specific drug compound that you treated cells in vitro with. And so you can generate this data or generate the microarray data that captures the biological differences induced by these compounds. And then leading from this, we can use these biological networks that have been constructed using the BEL language So essentially, we can overlay our microarray data onto the network, and then this can enable scoring using network perturbation amplitude scoring. And further from this, we can aggregate these scores and essentially look at the biological impact factor of how these perturbations affect specific cellular processes. And so this gives just a very general example of perturbations leading to enhanced cellular stress pathways, for example, as they're represented in the networks. Okay, so moving forward and just how to decode general BEL terms as they appear on the NBC website. And so a BEL term that are represented as nodes in the networks always appear in the same format. So essentially you have this colored box that indicates a specific function of the term. And then further from that, we have a namespace as part of the BEL term that indicates a database that contains an entity. So entity is really the protein or gene of interest, and this could also be a biological process. So moving on from this, this just captures AKT1 here being an example of a protein entity that we're interested in. And then the HGNC term represents the namespace that is a public database containing this entity term. And then, as I mentioned, the function describes an abundance or process related to this specific entity. And then further, how to decode a specific namespace term. And so there are multiple namespaces that are used throughout the NBC networks. And one example here being the HGNC database. And so what this database represents is just the collection of human gene symbols that in most cases will capture the entity that you're interested in reviewing or adding to the network. So again, this example being AKT1 is of course captured in the HGNC database. And so HGNC is essentially just the Hugo Gene Nomenclature Committee database. And this is a collection of all human gene and protein symbols, as well as all their related synonyms. But it also contains the official symbol for that gene or protein of interest that can be utilized in constructing a new BEL term. And you will see as you navigate through the NBC networks that a large portion of the nodes found in the networks will be derived from this namespace. And this is just a screen capture of, I know many people may be already familiar with this database, and this is just the navigation portal and how you search for genes on that website. Okay, and another example of how to decode a namespace. Another prevalent example in the networks being gene ontology, and this is represented by the GO term in the full BEL term. And this is illustrated on the right portion in the list of databases, just as the gene ontology biological process names. So this captures very general cellular processes, and this example being neutrophil chemotaxis. It could range from macrophage activation or DNA damage repair, or just more general process terms that can be incorporated into the network. And then again, this is just a screenshot showing the gene ontology website that's easily accessible, and you can search for any number of geo terms that can be also incorporated into the networks as new BEL statements. Okay, and another feature of how the terms are represented in the networks is the function term. And so there are a multitude of functions available that can be associated with entities and namespaces. And so these can range from protein abundances to the modification of these abundances. And a modification could represent a phosphorylation or ubiquitination term. And this also can capture processes and activities related to those proteins, as well as transformations. And we see that these functions are necessary to enable a representation of different aspects of a value. And here in this, the example on the right is the AKT1 protein being represented in multiple forms. And so moving from the top of the network, or starting with the top of the network here, we see that the protein AKT1 has a phosphorylation at 3-adenine 308, and another example being AKT1 phosphorylated at serine 4783 here. So these modifications are easily captured in the BEL language and can be represented as these modification terms. And probably the most prevalent instance or function in the database would be protein abundances, and these probably represent on the order of 80% of all nodes captured in the BEL encoded networks. And these are essentially just represented by the P function here attached to the AKT1 protein. And then moving from this, we can represent AKT1 as an activity term, and this example being the original protein abundance structure here that is wrapped with the kinase function. So this can represent the kinase activity of the AKT1 protein. And then moving from this and how these are connected to other components in the network, these can lead to additional downstream activities or processes. And so in this example, we captured the kinase activity of AKT1 has the effect of increasing the process of T cell activation. And so the T cell activation node is represented here again using the gene ontology namespace. And this is wrapped with the BP function to indicate biological process of the T cell activation. So this just gives a very general overview of some of the range of terms that you'll see in the NVC networks. Okay, and then how exactly the BEL terms are encoded in the networks and how the nodes are interconnected with each other using edges. And so based on a published experimental finding, the BEL terms represented as network nodes are connected together using a relationship term to specify a functional BEL statement. And so when we start with one example here in the top portion is a very generic this representation of a node in the network. And so this would be the function namespace entity basic structure. And this is connected to another node in the network using a specific edge type. And the edge types can vary based on the specific relationship that's been found in the scientific literature that connects these two. So this can range from increases or decreases, and these are represented as different edge representations in the network. So here moving toward a specific example would be the catalytic activity of the caspase-1 protein has the effect of increasing the protein abundance of IL-1 beta. And so here are screenshots directly from the NVC website. So you can, on any of the networks on the website, you can click on specific edges, and there's additional information displayed related to that edge, that specific edge. And so here would be one example of clicking on this particular edge for the catalytic activity of caspase-1, increasing the protein abundance of IL-1 beta. And so when you click on this in the network, you see that there are a total of 8 evidence lines that support this edge in the network. And these range from a variety of experimental contexts. And this could be primarily human and mouse support, but we also see specific relationships captured in human macrophages in lines 2 and 3. And there's also an example specifically from human chondrocytes for the last evidence line. And if you select any of these specific evidences that support the edge, you can also bring up this additional evidence detail panel. And again, this just repeats the relationship captured by this BEL statement. And it also has a quotation from the PubMed ID associated with this edge that gives more experimental context around this particular finding. And then here, as we see represented, we also have the species and the cell type captured for the context as well. And so when you review these edges in the networks, this is where you have the opportunity to approve or reject the associated evidence associated with a given edge. And I'll have more detail in the next few slides, but you're also able to add new evidence to support this existing edge. And so just moving back to the representations of specific edge types in the network. So one example would be one node increases the activity or expression of a separate node. And so this edge type would be represented with an arrow at the end. And then similarly, if you have a node that decreases the activity or abundance or process, this is represented by just the ball and stick model in the networks. And so this is a really clear distinction of how different edge types are represented across the network. And then furthermore, this is an example of directly increases. And so there are a number of instances in the networks where one node or the abundance of one node directly increases. And when we say directly, we just mean that the edge indicates the physical interaction between the two entities. And so in this example, the process of this node would indicate that it physically binds another entity to directly increase the activity or expression of the downstream component. And then similarly for directly decreases, again, this is indicated by a very bold edge line in the network. And so decreases would just have this ball and stick network model type again. And then another example of some edge types that you may see in the networks would be the non-causal edges. And so what this essentially represents is a relationship that's not supported by a scientific finding. And so a few examples would be protein to its respective, the activity of the same protein. And so these are connected using non-causal edges just because it gives more depth to the networks. But again, because it's the activity of the same protein, these are not supported by a specific literature finding. And another example would be a complex to a complex component. And so this could be one component of the NF-kappa B complex, for example, such as REL-B, would be connected to the larger NF-kappa B complex. But these canonical type relationships are just not supported by a specific scientific finding. And so these are also indicated as non-causal edges. And as we saw in the first example with AKT1, when it's connected to specific protein modifications of itself, so this would be such as phosphorylation of AKT1. So again, these are connected using non-causal edges, and these are not supported by a specific scientific finding. And basically, so the non-causal edges will not be reviewed during the challenge because the overall goal is to only verify those edges that have specific scientific support. Okay, so moving on to an example of how to add new evidence to a specific edge relationship. And so if you know about additional literature that supports a selected edge, you're easily able to go in and add this additional evidence as support in the network. And this also improves your standing within the leaderboards in the NVC community. And here we see an example of protein abundance of the IL-4 protein, leading to an increase in the transcriptional activity of the STAT6 protein. And so we go through the normal process of clicking on the edge to view the associated evidences in the NVC viewer environment. And when we click on this particular edge, we're shown that it already has 23 supporting evidence lines from a multitude of contexts. So these range from mouse fibroblasts and rat epithelial cells to additional human in vitro contexts. And so what we're able to do in this selected edge dialog box is click on the new evidence tab to basically enter these additional literature details into a specialized online form. And so here just is a screenshot of this additional evidence creation forms that would appear on the NBC site. And so for the selected edge, you would simply fill in this evidence form based on the literature reference you would like to add. And in this example, we found one publication, the title IL-4 and IL-13 receptors share the gamma-C chain and activate STAT6, STAT3, and STAT5 proteins in the normal human B cells. And so if we were specifically interested in how these two proteins relate to each other in B cells, this would be a perfect example of an additional evidence line that we could add that would further enhance the network architecture. So from here, we would just extract key details from the abstract or the supporting manuscripts associated with it and input these into the online form. And so the first input dialog here would be the reference ID. And this is where you would indicate the PubMed ID associated with this new literature. And then moving from that, we would indicate specific contextual information here at the bottom. And so this allows us to specify that it's human and the tissue is specifically B cells. And here are additional dialogues you could indicate if it was a specific disease context. And also, if it's done in an in vitro setting, we could indicate a specific cell type here. And then the quotation essentially just allows you to capture specific statements from the supporting publication that link these two entities together and better define the relationship that you captured as a specific edge. And so here we just captured the title of the paper, because that really captures the relationship between IL-4 and the STAT6 protein. And then once you click save and submit the evidence, and essentially at this level, it's incorporated into the network for other NBC challengers to view and vote on the evidence. And as I mentioned, you also get a substantial number of points for adding additional evidence to the networks. Okay, and another key tutorial for adding new BEL statements and BEL terms to a network would be how to construct an entirely new edge. And so here we see that this is just a screenshot representing the portion of the B cell signaling subnetwork. And here in this relationship, let's say we're specifically interested in the protein BONK or BLINK. And this is, as many people may be familiar, this is a key protein involved in transducing signals from the B cell receptor to downstream signaling events. And let's say we're specifically interested in this protein and we navigate to that in the B cell network. And so if we click on the protein abundance of BONK in the network, we're essentially we're allowed to zoom in on and see the precise relationships that currently exist in the network. And so as it stands now, we only have two edge relationships that are connected to BONK. So one being the protein abundance of SYK increases the protein abundance of BONK. And we also have the protein abundance of BONK increasing cell proliferation, as indicated by this biological process term here. And these two edge relationships are also indicated in the selected node dialog box underneath this panel. And so generally, if we're really interested in this BONK protein and we're left with the question of what if the network is missing a relationship with BONK that you're already familiar with. And so the way you would add an additional edge to the network here, and so let's say in this example, we found a specific publication from PNAS in 2001 that BONK mediates SYK-dependent BTK activation. And so of course we would want to enhance the network by incorporating this additional edge relationship into the network. And so the quotation here being, they've demonstrated that BONK mediates SYK-dependent BTK activation. And from the Create Edge dialog box, you would simply go into the Create a New Edge functionality, Then here we can specify that we found that the protein abundance of BEL and K directly increases the kinase activity of the BTK protein. And so in this specific example, the quotation we would include would read, we have demonstrated that BEL and K mediates SYK-dependent BTK activation. And an additional important evidence here would be that the phosphorylation depends on the interaction of BTK and BLINK. And so this direct interaction indicates that we should script this new statement as directly increases, because there is a physical interaction between those two proteins. And then so in here in this dialog box, we would simply indicate your source term, BLINK, indicates that it increases the kinase activity of BTK. And then once you click next, you're taken to step 2 in the online form. And this is essentially very similar to adding new evidence that we've already reviewed. And so in this example, we would just add specific reference information based on the publication that we reference. And so this being PubMed ID 11226282. And here at the bottom portion of the form, we would indicate specific contextual information surrounding this experiment that was performed. And then so in this specific example, the investigators looked at human HEK293 cells. So we would indicate this as part of the form. And then we would just incorporate the specific quotation from the literature that demonstrates the relationship between BLINK and BTK. And then once you click on next, this is incorporated into the network, and other NVC users can go in and review the relationship and then vote on that as well. So overall, that's a really quick tutorial of how specific BEL terms are encoded in the networks, as well as how you can use this knowledge to script new evidences and new edges to further enhance the networks. And some of the additional resources that you can use if you have very specific questions about the BEL language, or would like to see how this has been used in other examples throughout the scientific community. So one key example or one resource would be the Open BEL portal. And this is essentially a collection of all resources that the scientific community has pulled together and are using to just aggregate a lot of examples from the literature and promote better use of the BEL language. And then another resource would be the Open BEL wiki site that can be accessed through the wiki.openbel.org interface. And this contains a lot of additional documentation and training materials on the BEL language and how the BEL framework is structured. Then we also have a few discussion groups that you can easily navigate to and participate in. So this would be on LinkedIn as well as the Google group for Open BEL discussions. And then another really important resource is the GitHub repository. And so the Open BEL Consortium has posted a lot of resources on the GitHub repository that includes a lot of additional examples and documentation related to the BEL language and some of the sample knowledge that have been incorporated through additional research groups. So with that, I think I will switch over to a quick live demo and just illustrate a few of the key concepts that we went over in the webinar directly on the website. Okay, so here is the homepage of the network verification website, and I wanted to go through a few details about how the networks are, or how the BEL terms are specifically encoded in the networks. And so let me navigate to one example network, so neutrophil signaling. Okay, so here is the neutrophil signaling subnetwork, and we can see it's quite a large network. And so the basic representation here is that you can see the nodes are represented using different shapes. And you can see that as we demonstrated in the webinar, the protein abundance terms are represented very consistently as this teal-blue squares in the network. And another example would be the kinase activity terms being represented as the purple triangles. So in terms of navigating the networks and finding specific nodes and how they're encoded in the BEL language, so you can easily access the node list on the top portion of the network viewer panel. And so this gives a list of the 155 nodes that are found in the neutrophil signaling subnetwork. And essentially this is an easy way to browse through specifically how different biological terms are encoded in the network. And it's also really easy to zoom in on specific terms that you may be interested in. So here, if we select the kinase activity term of the AKT family, we can see that we're taken to a zoomed in representation with the kinase activity of AKT family sort of centered in the middle here. And this is connected to multiple different entities in the networks. And so on the edge panel on the right portion indicates that a list of all edges that are found connected to this AKT family term. And so this is also a really easy way to navigate through and find specific relationships in the networks that you're interested in. Let me reset the display. Okay, so one question that we have coming in from the Q&A is specifically how to filter the nodes in the network based on function type. And that's a really good question. So let me see. So if we go back to the node panel and access this from the top line of the neutrophil signaling network. So within the network viewer environment itself, you're not able to filter specific function terms in terms if you only wanted to see kinase activity terms within the network. That's not easily done within the viewer environment, but it is easy to navigate to that in the node list. And so you could easily select the kinase activity term, for example, and this gives you one method to filter through specific function types of the network. And so here we see a collection of all the kinase terms that are found in this network. And so this could be the kinase activity of IRAK4, and also captures a few AKT-related terms. And then similarly, you could filter this list by biological process. So this gives a list of 6 BEL terms that are associated with specifically biological processes that are found in this network. And so from this list, we can easily select, for example, the neutrophil chemotaxis node in the network. And so this allows us to zoom in and look at specifically what entities in the networks may influence neutrophil chemotaxis. So in this case, we see that there are a total of 26 edges that are found connected to the neutrophil chemotaxis edge. And this ranges from, so one example would be the protein of CCL3 increases the process of neutrophil chemotaxis. So if we select this example, we're provided with a list of the 4 evidence lines that are associated with this specific evidence line. And in this example, we see that there are two evidence lines from human neutrophils as well as 2 from mouse. that support this particular edge in the network. And so here it's easy to, if you click on a specific evidence line, to investigate the literature supporting this particular edge. And so in this case from human neutrophils, it's a publication from Cell Signal in 2005, indicating this relationship between CCL3 and neutrophil chemotaxis. So if you navigate to this PubMed ID and investigate this relationship further and to see if the experiment really proves this relationship, that's when you're able to approve or reject the statement as you see fit. And then for this existing edge in the network, so CCL3 increases neutrophil chemotaxis, you also have the option here of creating new evidence that supports this edge. Then this would be the dialog box that we went through in the webinar, where you indicate a specific PubMed ID and the contextual information that the publication would be supporting. So that was a really good question. Another question here is, what do the numbers represent next to the edges in the network? Okay, so that's a really good question as well. And so here in this particular example, so protein abundance of CCL3 increases neutrophil chemotaxis. We see that this edge displays in parentheses a 0 and a 4. Okay, so what these numbers mean is the first number is how many evidences have been approved by the NVC community. And so when we go through and vote on specific evidence lines, say if you approve this evidence, so that one vote would be counted in this first number, and that would be represented here. And so the first number is just the number of total approvals minus the number of rejections for a particular edge. And then the second number, so the 4 in this example, would be just the number of evidences that support that edge relationship. So here, again, we have 4 evidences, 2 from human neutrophils and 2 from mouse. Then if we zoom out into the larger network, we're able to see how these are represented across multiple edges. So in most cases here, there haven't been many approvals for these edges yet, and so the first number you see is most often 0, and then the second number just represents the number of evidences that support that edge. And so in this example, it would just be 2 evidences, and if we click on this relationship, for example, we see that it's 5 evidences that support the relationship between neutrophil chemotaxis and the CCL1 protein. So that's a good question. Then one other question we have is there a limit to the number of evidences that a particular edge can support? And the simple answer is no. So as you can see, as you navigate through the networks, there's a really wide range of evidences that support the edges in the network. So this could range from 1 or 2 to several dozen. So let me see. So in this case, we see that this edge has 9 supporting evidence lines. And yeah, the short answer is just no, there is no limit. So if you find an additional literature evidence that supports a given edge, you can certainly add that to the networks, and that goes toward counting toward your total score, and then your standing in the challenge. Oh, so another question we have here is how do the points work for different actions that you can take on terms and statements within the networks. So that's a really good question. And for that, I would direct you to the community tab on the website. And so if we navigate to the community tab, here's the opening page, the activity. And if you scroll or click on the leaderboards tab on the right portion, so here's a better explanation of exactly how these points are calculated based on specific activities in the network. And so the first example being if you create an entirely new edge, and if you create a new edge and it's approved by at least 4 other participants, this contributes 100 points toward your reputation score. So that's really a great way to quickly increase your standing if you'd like to get ahead in the score race. And an additional action that you could take to quickly increase your score would be to add new evidence. And so this would just be adding additional literature support to an existing edge in the network. And so if you add this evidence and at least 4 other participants vote on it, that contributes 50 total points toward your score. And then to a lesser extent, if you approve or reject evidences, so if you, as well as at least three other participants, approve or reject a given evidence line, this contributes up to 13 points toward your total standing. So certainly the fastest way to quickly increase your score is to find completely novel connections that are not already found in the networks and create these new edges based on new scientific literature. And so these are highlighted for the other participants to go into the networks and vote on these. And once that voting has hit at least 4, that's when you receive 100 points toward your reputation score. So that's a really good question. Okay, so with that, are there any additional questions that I can answer just based on how terms are represented, or how to create, or what specific actions mean in the network? Oh, that's a good question. Okay, so we have a question about adding new edges to a network. And so let me focus in on a specific segment of the neutrophil chemotaxis network. Let's say in this case, I selected CCL1, and the only edge that currently exists in the network would be this connection of increasing neutrophil chemotaxis. And so let's say we found another example that relates CXCL1 to a different entity in the network. And so what we can do is use this panel to create an entirely new edge. And so this is the new edge form that we covered in the webinar. And so what you can select here is essentially you can choose the protein abundance of CXCL1 as being the subject or the object term. So if you select the subject term, this is automatically populated in the online form. And then here you're able to select the relationship type as increases, decreases, or directly increases, as well as a number of other processes. But certainly the most common are increases or decreases. And so here there are quite a number of help functions available to help you properly script the BEL statement into the network. So here would be one example of a few of the helper dialog boxes. So here, in terms of generating a generic version of a BEL statement, this is just a basic representation of how it should be represented in the networks. And so this being the abundance of carbon monoxide, which is captured in the CHEBI database, increases the kinase activity of the MAPK14 protein. And so essentially, you just want to connect the subject and object terms using a selected relationship from the drop-down panel. And there are additional help functionalities available as you scroll through. So one being helpers to indicate the proper BEL function. And so this could help you specify whether it would be connected to a protein abundance or a biological process or a reaction term as part of the BEL statement. And then here are additional examples for the namespace identifier that would capture the entity term that you're interested in, such as TNF or emphysema. So the HGNC database, as another example, would capture the TNF protein, and within the gene ontology process database, it probably captures the emphysema database. The emphysema disease process. Then here, in terms of routine activities, so just additional help dialogues are here to help you navigate through creating a new edge and properly scripting these. And so when you have, if you have CXC01 increases, let's say it's AKT again. So if you start typing just the P function to indicate protein abundance in the object term dialog box, that's when you, an additional help functionality is introduced here at the bottom portion of the panel. And so this could basically gives you a full listing of all of the available functions, namespaces, as well as a few of the entity examples that can help you better script the statement. And so here we would just select, if we scroll through the function box, here we see the P corresponds to protein abundance, of course. And then as we further type along and introduce the object term, we can type the HGNC database. And here we can type the AKT1 protein. And then so here when you type AKT1, the form dialog box will automatically search through the HGNC database and find the most relevant terms that it thinks you're trying to enter into the statement. And so here one example would be just AKT1, as we've already introduced into the statement text here. And then another term found in the HGNC would be AKT1S1. And so the help functionality really tries its best to help you fill in the terms and construct the best statement possible when adding to the networks. So here, if we just complete the object term, for protein abundance of AKT1, and so that's when we progress through to the step 2 and add the specific evidence to support this edge. And so here we would just select PubMed ID, then indicate one specific example. So just entering a generic PubMed ID in this case. And then we also have the capability to specify specific context. And so if we found that relationship existed in human neutrophils, we simply indicate neutrophil in the tissue component box. And if this is found in a specific disease state, such as asthma, let's say, we can indicate that in the context as well. And then here, the only other required text is for you to enter the quotation. And so that would be sourced directly from the associated scientific finding that you located. and then just enter it into the dialog box. And once you do that, you would just click next. And then that's when you have a confirmation of all the evidence and statement that you've entered. And then here we can just submit this as a new edge in the network. And so it's a relatively straightforward process, and just the edge creation tool really helps you to construct the best statement possible and locate the appropriate namespace related to the terms you're trying to enter. So that's a really good question. Okay, so I think that wraps up all the questions that we have from the participants today, and thank you for really good questions as part of the live demonstration. I think that will that was a great help for everyone to better understand how these terms can be entered into the knowledge of the networks. Okay, so with that, I'll just end with and thanks everyone for attending today. And I would just direct you to the SBB Improver website to participate further in the challenge, and also to find additional information related to the specific concepts that we covered today. Okay, so I think that's it. Thanks everyone.